our next speaker is Jennifer Smith from RGD, from the Rat Genome Database, and she will be talking about OntoMate, a text mining tool to facilitate curation at the Rat Genome Database. So this is a short talk, right?
It is a short talk, yes. So hang on. We're gonna go fast. So I'd like to thank the organizers for giving me the opportunity to talk to you about OntoMate, our text mining tool that facilitates curation at the Rat Genome Database. And I'm going to mention right up front that I do have a poster. It's H23 if you'd like to get more detail, because I'm not going to be able to give you a lot of details here. So RGD is a cross-species platform for translational research. RGD presents data for 8 species: rat, human, mouse, chinchilla, bonobo, or big pygmy chimpanzee, 13-line ground squirrel, dog, and pig. And if you'd like to know why those were chosen, you can come talk to me. Species are chosen because they're models for human disease, and data objects such as genes are annotated to multiple ontologies. In the case of genes, they are annotated to disease, gene ontology, phenotype, pathway, and gene-chemical interactions. And for those of you who know about curation, curators have to go out and find the literature that they're going to be able to curate, which is a problem in this case because PubMed contains more than 1.6 million records for rat, and even more than that for human disease, but only a subset of those can be actually used for curation. So we needed a solution that would help the curators to more reliably and more quickly and efficiently be able to find the literature that they needed. So we developed the OntoMate system, and this is a diagram of the architecture, and I apologize, some of the text is probably pretty small, but you can look at this on my poster. But I'm going to hit the highlights right here. So abstracts are downloaded from PubMed into a Hadoop H-based NoSQL database. The original download was everything that PubMed had, all of the abstracts in PubMed at that time. And then since then, incremental updates have been done every day to make sure that OntoMate is up to date with what PubMed has. And then XML parsing is used to extract the title, the authors, the text, publication date, and citation. Then existing bio-NLP tools were used to extract and match terms from multiple ontologies, as well as gene symbols, species, and mutations or variants. And we did not actually develop any natural language processing tools. We used the ones that were available. So those produce tags, and those tags are added to the article database, and then titles, authors, abstracts, ontology terms and IDs, and so forth are indexed using Solr. The UI for searching using OntoMate was integrated into the RGD curation software, and it's actually a standalone tool of OntoMate is available on our website if you're interested in checking it out. But I'm going to talk about the instance that is integrated into our curation software. So the original workflow for curation at RGD was that curators would manually add a gene and term-specific query string into the PubMed interface, query PubMed, and then manually transfer PubMed IDs, genes, ontology terms, and so forth to the curation tool and do their annotations from there. And this is actually a string that was used for searching PubMed to do urogenital and breast cancer curation. And you can see that it's very long, and it's a very complex query that was trying to grab everything that a researcher could have used to refer to a particular disease and get the ones that we needed and not get the ones that we didn't. By contrast, the current workflow curation is that curators will manually add gene and upper-level ontology terms to the curation tool, query OntoMate, and then use OntoMate hyperlinks to add the references, extra genes that they might be able to curate from what they're getting, and specific terms to the ontology tool. And this would be a set of terms that would get entered into OntoMate now. So you would enter a gene ortholog group because we annotate across to orthologs, and then one or more upper-level ontology terms such as urogenital neoplasms or breast neoplasms. So once they have done their query, this is the kind of result page that comes up. There's a lot of information on here, and I'm not going to be able to go through all of it, but basically the UI is designed around the way RGD curators do literature curation. And the features show that a paper has already been curated or is not curatable for various reasons. Disease and organism terms are shown by default so that the curators can just scan through and see if there are diseases they're interested in. And the full abstract and additional terms from other ontologies open up with a single click, so the curators can easily get to the more complete information. So the advantages that OntoMate has given us is that OntoMate leverages the structure of the integrated ontologies for its searches, and also leverages the information associated with the entire ortholog group to find references even when researchers use non-standard nomenclature. OntoMate is designed to let curators see at a glance what a paper has to offer, and it's also designed to show curators if a paper has already been curated, if it has been read but not curated, or if it can't be curated at all. And OntoMate is designed to allow curators to see additional curatable data in a paper that they might not have thought to look for. And the biggest advantage is that OntoMate is fully integrated into RGD's curation software, reducing error-prone actions like copy and paste, and eliminating the need for curators to use multiple windows to try and locate articles, browse ontologies, see what's been curated from a particular paper, and so forth, outside of the curation tool, and then have to copy that stuff into the curation tool. So in terms of future directions, RGD's PhenoMiner tool requires curation of specific quantitative phenotype values, specifically those from tables or from texts of the results, rather than from graphs or charts, because you can estimate what a number might be from a graph or a chart, but you can't get a specific value. And finding such specific data can be difficult and time-consuming for curators, and actually is quite difficult and time-consuming. Such data is often located in the supplementary materials in more recent papers, and in many cases, it's not available at all for older papers, such as those that describe the development of a rat disease model. And so, yes, OntoMate is already indexing the ontologies used in the PhenoMiner curation, and so our goal is that we will improve OntoMate to tag not just abstracts, which is what it's doing now, but full-text articles and the supplementary material for those articles, and be able to tag them as containing putative data tables suitable for PhenoMiner curation. And with that, I would like to say thank you and acknowledge the group and our funders, and invite you to come and talk to me and get more details and see more pictures at poster H23. Thank you.
So when you talk about some papers not being curatable, I assume that means that the papers may be talking about something but not actually showing it, so it doesn't have the evidence. Are you then making use of citation information? Because I guess often they would then refer to the paper you would actually want to curate.
Yes. Yes and yes. A lot of times a paper might not be curatable because it appears to be talking about, say, rat, but when you get into it, it was indexed as talking about rat because they used a rat antibody, or they're using cell lines and the actual data is for a mouse cell line or a human cell line, not for rat. So a lot of times, it's the species problem. But the curators do go back, they do use the references from papers to go back to the original papers to get the actual data, if that's available. As much as possible, I mean, the time that it takes to follow something all the way back can be prohibitive. So, thank you. Okay.
Thank you. Okay, so our next talk is a combined